Hello, everyone. Hello to all the listeners in the live webinar right now, or the people who are watching on YouTube. This will be our second guest speaker. Before we get started, I'm going to give a quick overview of what Pathways is, just in case you're new. So Pathways is a nonprofit organization that provides the opportunity to middle schoolers and high schoolers to learn about all sorts of different careers and help clarify their career path through learning through two main methods that we offer. The first main method is the first service that we provide our guest speakers. We have them every 2 or 3 weeks through Zoom webinars. And in these Zoom webinars, such as this one right now, you get to learn about how these guest speakers got to their career, how they explore different careers. And you also get to learn about these careers broadly and specifically. So in the example today, you can learn more about project management, but also you can specifically learn about cancer research. The second service we provide are research groups, which happen in the Discord server. So we form small research groups, and you can, have research groups with your friends or people that you don't know who are on the Discord server. And then we have services for you to schedule times to actually research different careers with other people inside of the Discord server. So we do this because I've talked to a lot of high schoolers, and I know that it's hard with our busy schedules to have the motivation to just get up and start researching careers out of nowhere. It can be really hard to find a starting point. So these groups are a good way to stay motivated and stay disciplined and be able to talk about what you learned through these different careers. So without further ado, Ms. Mathangi, if you want to introduce yourself, that'll be great.
Thank you so much, Vikram. Hello, everyone. I'm very excited to be here. Thank you so much for the invitation. My name is Matangi Tyagarajan. I'm going to tell you a little bit about myself, what I do, and I'm more than happy to answer any questions at the end of the seminar. My name is Matangi Tyagarajan. I'm a proud mother of two kids. My son, an 18-year-old, is a freshman at University of Maryland, majoring in computer science, and my daughter is a 7th grade, a middle schooler. So I know exactly how you all feel because we just went through this whole college process, and I have a kid in middle school, and it's amazing that Vikram has founded this organization and a community service for sharing information about career paths with many of you. So kudos to you, first of all, and thank you so much for inviting me. I am, other than being a mother, I am a project director for a program called CPTAC. I'm going to talk a little bit about the program as well as what led me to my career path. Is it okay if I share my screen now, Vikram?
Yep, that will be perfect. Thank you so much.
Are you all able to see my screen?
Yep, looks great.
I don't know if I shared it with the, I'll share some, yes, okay. Okay, so as I said, I'm a project director for this program called CPTAC. I come under the Biospecimen Research Group for the Frederick National Laboratory for Cancer Research. We call it the FNLCR. FNLCR is an FFRDC, which means it's a federally funded research and development entity that is owned by the government but run by contractors to support the operations of the government, which in our case is National Cancer Institute. Today, I'm going to talk about my introduction, my education, and what I do in my job. I was born in Chennai, India. I was schooled in Jawahar Vidyalaya, JV, and my high school was in Fatima Convent in Chennai, India. I moved to the US in 1998. While I did my bachelor's or undergrad in India, majoring in chemistry and minoring in biology, I did one master's back in India, which was a major in biochemistry with a minor in genetic engineering, which was my first real exposure to genetics. After coming to the US in 1998, I did another master's in biomedical informatics with a specialization in genomics and proteomics. Way back then, I had kind of narrowed it down as my field because I had a natural instinct as well as tremendous interest in this field, which was very, very new at that time, especially when combined with bioinformatics. Over the course of time, I've done many, many certifications that made me better in learning as well as in the job that I do. I would also talk about few mistakes that I made. One mistake that I did was, since I was so interested in science back in high school, I didn't pay enough attention to math. I kind of dropped math at 10th grade and moved to a pure science track of just focusing in chemistry and biology, which were my primary interests. Even though I knew what I wanted to do, it was a realization later that I should have pushed math a little further down, at least until my first year of college. Job experiences, I've been in the field for 20 years now. My first 10 years, my formative years, were primarily technical positions. I worked for this genomic research institute called the Institute for Genomic Research. It's one of the pioneering institute that focused on genomic research way back then, that eventually became the Human Genome Project through Celera Genomics. And it was then renamed as the J. Craig Venter Institute. J. Craig Venter, who was part of Celera that led the Human Genome Project, was the founder of the institute. I spent a good 10 years there, starting as an analyst and then climbing up the ladder as a team lead, and then was leading a group to analyze genomic data. So a good 10 years of real hands-on analysis on genomic data really, really laid my foundation in the field. After a good decade, I moved on to FNL-CR, where I now work, and I've been there for almost 10 years now, supporting National Cancer Institute on various programs, primarily genomics, and eventually I moved into proteomics and the combination of proteogenomics, which is what I will talk about. So where does my career categorize? It's categorized as a scientific program management. Basically, I manage these programs. I have a group that does quite a few functions, and that eventually leads to scientific papers as well as goals that we set at the start of the program. And my focus is primarily on genomics and proteogenomics, even though I've been a subject matter expert on many other projects and proposals. These are the programs that I run, but there are other programs where I participate and provide some scientific support. Some basics about my field. So I'm sure you all would be aware of what a genome is. It's literally the set of genes. Studying the set of genes in an organism is a genome. And the field of studying the genome is genomics. And similarly, proteome can be considered as a counterpart, where it's the study of a set of proteins in any given organism. And a combination of genome and proteome, because genome is like the blueprint of life, and proteome is what gets that signal from the genome or the gene that does all other functions in the body. So the combination of genome and proteome is basically proteogenomics. And what I'm going to talk about once we look at a video now is to see how we can use something as powerful as studying the genome and the proteome of a cancer cell in understanding how we can leverage that to better diagnose, treat patients, as well as prevent down the road. So this is a very, very new, powerful field that has been making a lot of headlines. And let me quickly open up this link.
Proteogenomics research on the frontier of precision medicine. The fields that we currently consider next-generation molecular medicine, including genomics, proteomics, miRNAomics, microbiomics, and epigenomics, all share the same foundation. That foundation rests in omics, which is an informal term used to describe the comprehensive study of the biological components of a cell at the molecular level. Take genomics, for example. Genomics is a comprehensive study of the complete set of genes, DNA, in an organism, which we refer to as the genome. Analyzing the genome of a patient's cancer can reveal information about how to best detect, diagnose, and treat the patient. For example, a patient's cancer may have a DNA mutation that makes it especially sensitive to a drug. Although studying a patient's DNA can provide a lot of information, it doesn't give us the whole picture. The reason is that in addition to DNA, there are many other molecules that contribute to cancer biology, such as proteins. Proteomics is the comprehensive study of the complete set of proteins in an organism, which we refer to as the proteome. Proteins are built from DNA. You might say that DNA is the blueprint for life, and proteins are the tools that make living systems work. They play a major role in the daily functions of both healthy cells and cancer cells. Analyzing a patient's cancer proteome can also provide information about how to best diagnose and treat the patient. This is because proteins carry out almost all of the functions within our cells. If a protein is altered, a cell could begin to multiply uncontrollably, potentially leading to cancer. This is why almost every clinical cancer treatment targets a protein. Understanding how proteins influence cancer biology is critical to developing better diagnosis and treatment strategies, as they can add clarity to how a patient will respond to a particular treatment. In the past, researchers often studied patients' cancer genomes and proteomes separately. But in 2016, research teams combined the comprehensive analysis of patients' cancer genomes and proteomes. This integrated approach of proteomics and genomics is called Proteogenomics. These studies showed that separately, genomics and proteomics provide a partial picture of cancer biology, but studying them together produced a more complete, unified picture. Researchers are hopeful that proteogenomics may improve our ability to prevent, diagnose, and treat cancer at the molecular level. Proteogenomics research on the frontier of precision medicine. U.S. Department of Health and Human Services, National Institutes of Health, National Cancer Institute.
So I'm pretty sure you would have had an overview or a basic overview of what is a genome, what is a proteome, and how we combined it to make it proteogenomics and why this is important for cancer research. Just to give you some timelines of what this is about, when NCI, National Cancer Institute, was thinking about creating an encyclopedia of all the genomes as in cancer is not one disease. There are many different cancer types based on what they impact. There's breast cancer, ovarian cancer, brain cancer. There's so many different cancer types. And even within each of those cancer types, there are many grades, many stages. Everything is so different that there is no one size fit all. They have to be extensively studied. So first of all, if we can understand the intricate problems of each one of these, then it's very difficult to come up with solutions. So characterizing it like an encyclopedia of all these cancer types was a national initiative that was started in 2006, what we call as the Cancer Genome Atlas, or TCGA. It's a flagship program, a national initiative for 10 years to practically characterize at the molecular level of 33 different cancer types from 20,000 patients. And the goal is to diversify these samples so much that we can understand what these are at the genetic level, so we can better do a better job of coming up with drugs. So when this was done and it became a huge success, NCI's next step was to move it one level above and do it at the proteomic level. So CPTAC, the Clinical Proteomic Tumor Analysis Consortium, I would say is the counterpart of TCGA at the protein level. It is not like we have to characterize every little thing at the proteomic level. We just have to do it for those cancer types where it matters, where proteomics will add additional value, additional information, where it's feasible, because we are not doing this in parallel. We are doing in combination with the genomics, the already done effort of TCGA. So we selected 23 different cancer types from 5,000 pages. The goal was to prospectively collect these samples, taking a lot of lessons learned from the original Cancer Genome Atlas project, which I was part of for 4 or 5 years, and implement it at the protein level and identify what we could not identify from the genome that must be done at the proteomic level. How can we take this to the next level of translating it from bench side to bedside? So the phase 1 was started initially just as a technology assessment of how we can implement this in proteomics. That took a while, but that really solidified the technology that this is really feasible and extremely valuable. So a pilot of 3 cancer types, ovarian, breast, and colon or colorectal, was started in 2012 to 16 that proved to be a huge success of identifying not just interesting subtypes in those cancer types that could not be identified at the gene level, but also very interesting biology that was originally missing just from studying the genome. This then expanded to another 10 tumor types between 2016 to 2020 that I was a lot involved in. And now we are entering into the next phase of rare tumor types, pediatric, and so many other newer tumor types that can eventually lead these into early stage clinical trials. I would highly recommend going to all these websites. There are publicly available data from the analysis that's been done if you are interested. The Cancer Genome Atlas data is available at GDC, and the proteomics data is available at PDC. So we already saw why this is important to combine these different omics technology. We get a lot of imaging data that is like MRI scans, that is used for the actual diagnosis of these cancers. And then we have these genomics and we have these proteomics. Individually, many of these things have offered a lot of insights as to what are those driver mutations, what caused the cell to change, why did it grow uncontrollably, which oncogene got triggered and why, which tumor suppressor got suppressed and why. So a lot of these questions are always asked when a patient comes in with cancer, and it has been well proven that there is no one technology that's going to give all the answers. We have to be able to combine these multiple technology. Sometimes they confirm each other, sometimes they contradict, but they always provide a little bit more to that missing piece in the puzzle that's going to help a physician or an oncologist to better understand that cancer and better treat the cancer. So what is my team's role in this whole program, CPTAC program? My team's role is to procure the biospecimen, the cancer tissues that are needed for these characterization and downstream analysis, process them appropriately, characterize them, and gather all the associated clinical data, which is as important as the samples, and provide it to the researchers for them to draw conclusions from. So we are at the front of this entire pipeline where my team works with several entities, what we call as tissue source sites, where we work with clinicians, hospitals, oncologists in identifying patients, make sure that they consent to this research, and they give a sample from that surgery, make sure that they are all being collected the proper way. And these samples, along with the clinical data, comes to a place called the BCR, which is Biospecimen Core Resource, which can be considered as a central hub. We want to make sure that when we create a giant encyclopedia with all the information, then we have to make sure that they're all apples to apples. They're all collected in somewhat similar manner. They're all followed a specific protocol and an SOP. A lot can vary just in how a sample is collected, right from when the sample is picked and how it is stored and how it is shipped, under what temperature, and how these are processed. So making sure that these are collected the way it should be collected. And as I said, cancer is no one disease. It's a very complex disease, and there are multiple cancers, and each one of them have a separate protocol and SOP. There is no way we can collect lung cancer tissue the same way as we collect endometrial or ovarian. Each one of these are slightly different or completely different, and we have to make sure that they are all collected the way they should be collected, and they are processed and qualified both pathologically as well as at the molecular level, shipped properly and to the characterization centers, done genomics and proteomics on, and combine all these data together and put it into a database, and then give it to the analysis centers as well as the translational centers that are going to interact with the database and get information that would help them in identifying scientific insights that are eventually written into publications and are published and are shared with the research community. So specific responsibilities that we are in charge of is to collect these biospecimens as well as the clinical data. There are a multitude of functions that we follow in order to do this. First of all, we have to subcontract with these tissue source sites or hospitals. Sometimes they are hospitals directly, sometimes they are academic institutions that have access to a laboratory or a hospital. Sometimes they are CROs, contracting research organizations, that act as like a middleman, because we don't want to cure cancer just for US. We want to make sure that we get samples from all over the world. We work with, my team works with about 40 different tissue source sites. Half of them are domestic, half of them are international from across the globe. And we try to make sure that we have enough diversity in our cohort. We make sure that we get all kinds of samples, whether male, female, age group, the race and ethnicity. We want to make sure that we have a good, because these are considered as like an encyclopedia. We want to make sure that we truly understand the biology of these cancer, which means we have to be as broad as possible, as well as bring in a lot of standards in how we collect these cases. So it is extremely challenging to work with international sites, because even their medical records may be in a slightly different language. They have to be translated. There could be different regulations within their hospitals in sharing samples and sharing data. There could be customs issues. So making sure that we put in these appropriate contracts with these entities from various tissue source sites, making sure that they all follow their regulatory compliance, making sure that the patient is well consented, they know what they are giving that sample for, and they are okay providing the information. All this information is de-identified, making sure that there is absolutely no personally identified information that can come out. All those things become the responsibility of the team to make sure that these samples are collected. They have to be collected within 30 minutes, what we call as the ischemic time, from when a surgeon cuts the blood supply to the tumor cell, from when those piece of tissue. And since we need to do both proteomics and genomics, we need enough mass. So we need to make sure that we get that piece of tissue. They have to be immediately flash frozen and transported, and they have to follow a very strict specific protocol. So making sure that these are collected and procured the appropriate way. And then we wait for these to like group up, and then we ship it to the central hub, our BCR, the Biospecimen Core Resource, where we first QC them. We make sure that what we thought the sample should be, whether it's the right grade, whether it's the right histologic type, whether we have enough mass, are there any issues with the sample? Is it of the highest possible quality? Because this is the first starting step for this research. The highest quality we can provide from these samples really goes a very long way in the meaningful science that can come out of this. So making sure that these are appropriately handled and then processed and qualified. We work with many pathologists. Some are general pathologists, and then we go to experts for each of these tumor types. We want to make sure that a lung specimen is reviewed by a lung specialist and not by a general pathologist. So we review these slides and then we qualify them. And then we do the actual extraction of DNA and RNA because we want to do both genomics and proteomics from the same sample. So if there are multiple segments, we put them together and we pulverize them and then we aliquot them, because when we want to compare and contrast, we can, if we take one piece of tissue and do genomics and another piece of tissue and do proteomics, cancer is such a heterogeneous disease. It can actually be different between cell to cell, and we might not be able to say what is right. So making sure that we are indeed comparing apples to apples, we have to process these in a specific way and then send it to our proteomic characterization center as well as genomic characterization center. The technology that we use for proteomics is mass spectrometry, and we have a lot of new technologies that's coming. And for genomics, we do the DNA and RNA isolation, and we go through DNA sequencing, whole genome sequencing, whole exome, as well as we do some methylation and other omics technology. And for RNA, we do transcriptomic sequencing, which includes a total RNA as well as microRNA sequencing. So as you can all see, I mean, there's a lot of data and information coming from these. And we have built an in-house portal to monitor this workflow from the start of when a patient consents to us getting this data out. Everything is tracked through an informatics portal that is built in-house, where at any point I'll be able to log in and see where a specific sample is. And there will be a lot of key decisions that need to be made, whether we can ship the samples or not. Are there any issues? So constantly we have project managers in the team who monitor these through these portals, the actual movement of these samples throughout, because a lot of critical decisions have to be made. And these are very, very precious samples. They are not that easy to get at all. And it is really our responsibility to make sure that we get these samples and the accompanying clinical data. So the clinical data we develop these case report forms. These are basically forms. For each patient that we collect a sample, we at least collect 250 data points, right? From whether the patient has any smoking history, alcohol history, whether the patient had any occupational exposure, whether the patient had any family history of cancer. So right from basic questions like these, we at least collect 250 or so data points from each of these samples. I mean, most of the time it's through the physician I mean, we don't trouble the patient. We make sure that we keep the interaction as minimal as possible. But the important thing here is that those accompanying clinical data provides a lot of insights to what we want to do downstream. So it's as important as the sample itself. And making sure that if there are any inconsistencies between the data, we have a team of data management people who reviews for and checks for the correctness and completeness of data. We get statistical power only if we have as much data as possible to be able to make these inferences. And we also have like a quality management system that across the board will make sure that everybody follows the same SOPs and documentation. If there is a non-conformance event, then we have some kind of a corrective and a corrective plan when something like that happens, an incident happens. So a lot of these things go hand in hand, and there's a lot of teamwork that is involved. And overall, this is what we basically do put together as a program management. So as you can see, this is a really exciting time for the convergence of science, from the images that come out of these samples, from the big data. The proteomics as well as genomics are really large data that comes out because we are studying every little information in those genes and proteins. And the actual pathology images and the information that comes from these clinicians, oncologists, statisticians. So combining all these together is what the downstream analysis teams need in order to make their scientific inference. So results, as you can see, our proteogenomics program, I mean, this is just few of the many really top-notch marker papers that we have published in the past couple of years from when CPTech started. And it is extremely fulfilling to see that cancer is such a deadly disease, And we are extremely thankful to all those patients who agreed to give a little bit of their sample from the surgery towards this research that is going to benefit the mankind down the road if we can make better drugs based on all the information that we are getting from at the molecular level from these projects. So where does we fit in again? How does it is all teamwork again? As you can see, we are the starting point in terms of getting these samples on the clinical data, but most of the time we have to interact a lot with the research community because as you might all know a little bit about program management or project management, the way we plan things really matters when it comes to end results. But how do you plan something where there's a lot of moving target? I mean, this is research, right? Halfway down the road, we will figure out that the samples are behaving slightly different, or we thought we had this much tumor nuclei in it, but that's not the case. And halfway through characterization, we might identify something completely different. There are so many things that can change, that can go right or wrong. So based on that, we may have to make some quick decisions. So interacting with the research community, interacting with the translational arm, interacting with the analysis teams, constantly giving them information of what we are collecting, and getting feedback from them and feeding it back to our sites that are collecting samples. We can say, well, we've collected these samples, but it turns out that we are not getting one histologic type enough to be able to say anything meaningful. So how can we get that particular histologic type, more of that? Things like that. And sometimes things fail, and then we have to go back to a repeat sample, or sometimes the patient might get a recurrent sample, and that can provide a lot more biology than their original primary sample that they donated to the program. So a lot of that, it's not just like, we collect, we turn it to them, and then we don't know what's happening. We continuously work with all these disease working groups and the oncologists and the researchers, and make sure that their feedback comes back to us in terms of providing the high quality samples that they need to continue their research. So a lot of teamwork is involved. So I'm happy to, I know a lot of questions came, which I really appreciate, because now I really know what to tell. What training and education was required for me to do my job? Well, first of all, my passion for science. I cannot tell you all enough how much I love science right from when I was a kid. Biology and chemistry interested me most. I didn't know anything about oncology or research. Those are all like science that I picked up down the road. But as as a high schooler, I was fascinated by biology, and I originally wanted to become a doctor, which I couldn't. It was a highly competitive field back in India. And it was my disappointment, but nevertheless, that never let me down. I somehow wanted to contribute and be in healthcare in some kind of a meaningful way. So I did my bachelor's in chemistry and biology, as you all saw, and then I switched to a hybrid approach of biochemistry with genetics. And then I moved forward with biomedical informatics with genomics and proteomics. So it was a very systematic approach of getting into healthcare research, but utilizing what I was good at, what I was passionate about. Several technical skills that I learned down the road, not when I was in school or in college, were definitely biospecimen handling, omics technology. I did do genomics and proteomics back in college, but it was very minimal at that time. It was the field itself was pretty primitive 20 years ago. And of course, informatics, I'm not a hardcore programmer, but I've always been fascinated by technology and using technology, especially in healthcare. So that has always inspired me and intrigued me. I always read a lot about it. I take some courses and see how I can integrate the two together. And I'm all about big data. Every decision that I've ever taken in my little team is all data driven. What site to go after, how many more samples do we need? Everything is driven by data. And this is the data science era. So really leveraging those skills really helped me to do my job well. The management skills, again, project management best practices. It is really important. And in a research environment, especially, you can easily get pulled in different directions. Somebody would want this and that halfway down the road. So if we go out of scope, then it could really uncontrollably go in many different directions. So making sure that we have that big picture in mind, keeping the project on scope, keeping the project on budget and on schedule, making sure that we deliver these samples and the clinical data on time goes a very long way. Anticipating, identifying risks that can go wrong, especially during COVID. We had a lot of issues because most of the hospitals were repurposed for COVID, and a lot of surgeries were scheduled and then canceled. We had issues transporting and shipping samples that were already collected. They were held in some location that we couldn't get hold of. So a lot of these things happen, and that we have no control over. So anticipating these risks and coming up with a mitigation plan and making sure that we implement when the risks get realized is definitely a very important training that I learned. And these are all like real skills that I took, either courses or education, but there are also a lot of soft skills that I've learned over time, which includes communication skills, problem solving, critical thinking, decision-making, writing skills. No matter how well you do in a project, if you can't present that work in a proposal or a report, then nobody knows about it. So making sure that you effectively write these results and communicate information through effective writing is really important. And of course, teamwork. It's really important to be a good manager of people, so we can keep the people motivated and driven, and that goes a very, very long way. So if there is one thing that, there is no training or education, but I've learned from watching people and from trying new things, is definitely that kind of a leadership, where I'm definitely not the kind of person who tells people what to do. I do it with them. Getting your hands dirty on the actual work goes a very, very long way. There's a saying, I strongly believe that good engineers become good managers, which means if you don't know how exactly, you don't have to be an expert in everything, but if you don't even make an effort to learn how something is done, then it's very difficult for you to tell your team how to do it. So really rolling up your sleeves and getting the work done is something I have done a lot. And that has really made me have an awesome team. I'm very, very proud of my team. And everybody is a very, very talented group of individuals. And that definitely drives the mission forward. What led me to this job is simple love for science and technology. I'm pretty sure, I wanted to be a doctor. If not, I wanted to be somehow involved in healthcare, and technology really fascinated me. I was just on that period of era when the Human Genome Project came in 2000. So there was just no turning back. That is it. That was the trigger. And I simply went after it. And I definitely was lucky to have some right opportunities that put it all together. But no matter what troubles and issues we have in the project, at the end of the day, when we see a paper, when I see a drug getting approved, when I see these numbers go down, it gives you that satisfaction. It gives you that feeling that it's all totally worth it. So that kind of feeling is something you can get only if you're truly, truly passionate about what you do, because no job is easy, but if you want to really excel in it, you have to put your heart into that job. Don't do it just for the sake of doing it, do it with all your heart, with all your passion. What I like about my job is, of course, opportunity to work on cancer research. It's no ordinary thing. It is such an honor to work on cancer research because it's so fulfilling and it's so interesting, cutting edge science truly, and technology. And I truly enjoy problem solving. Sometimes I crave problems, I would say, because it gives me a sense of satisfaction when as a team, we solve problems and we really truly back it with data and true investigations. It's very, very fulfilling. And I like to present information to different kinds of audience and stakeholders. That's something I consider myself good at. And I like challenging problems, and I truly sit with my team and brainstorm, and we come up with some long-term solutions that have come a very long way. And we have a reputation for doing that, especially some troubled, hard to deal with projects. We worked very hard and we've come a long way. And I also like new initiatives and business development. I always contribute as a subject matter expert. So those are things that I really enjoy. And I really look for opportunities to integrate IT with science. Even though I'm not a hardcore IT person, it really fascinates me when science can be leveraged when combined with informatics and technology in this day and era, which is all bioinformatics is about. And building and mentoring teams, and I love to negotiate. What I don't like, I would say, being in a government project, it's, there's a requirement to do a lot of reporting, which can sometimes be overloading, but there's just no two ways about it. I'm not a big fan of finance tracking. Having, as a project director for a program of this big, I have to do a lot of finance tracking. This goes back to me not doing enough math in high school. But I'm very, very thankful to a very able team of project managers who really back me up on the finance tracking, which I generally don't enjoy doing, but I still have to do it anyway. Not a fan of formal meeting notes. I'm a bullet points person, but that doesn't go very well sometimes in a government setting. You need to write some formal notes and documentations. And sometimes certain policies that are completely out of our control can really slow things down. And there's just no control. We have no control over it. It's just the way policy works in a government environment. And we simply cannot bypass that, especially when it comes to something this critical. And of course, since I like to be involved in everything, I sit in everything, but that can also cause some meeting fatigue. Those are the things that I do not like that much. So what courses in middle school would help a student? Well, I would really like to spend a little bit of time in this, because I believe that a very strong foundation on basic sciences is extremely important, because your foundation should be very strong. I see a lot of people narrowing themselves to something very niche and unique very early on, while it's not a bad idea to do that. If you're interested in, I don't know, artificial intelligence or genetic engineering, you still need that biology, you still need that chemistry, you still need that math, you still need that computer science. So you have to make sure that you lay your foundation very, very strong and then narrow it down. Because if you narrow it down too soon, unless you're 100% sure that that is your field, you can easily pigeonhole yourself into a field. And after a few years, for some reason, if you don't like it or it's not working, then you've already gone too far into a very narrow area that it becomes hard for you to turn back and come back to the basics and then identify what else is out there. So I would highly recommend. I know things are changing a little. People reorient after doing something very specific and niche, but you can never go wrong with the foundation, the basic science. And the stronger you get, the more robust you get your understanding. Really make sure that you understand the science. Never be afraid to ask a question to your teacher. It's not just about getting grades. It's about really, truly understanding what you're learning. Go back again and again, because trust me, I still go back to my 10th grade, 12th grade book, even though I've been in science for so many years, because those foundations are extremely important for you to do well down the road. People in your team will respect you only if you truly know your stuff. I mean, I know a lot of people also do management degrees too soon, but at the end of the day, what do you manage? You're going to manage some business, engineering, healthcare, something. If you don't have that underlying understanding of the technical details, then your management is really based on what others tell you. You may not really know if that is true or not. So really spending some time on the underlying engineering aspects or the technical aspects or science, whatever it is at your field, please make sure that you spend enough time. Don't jump hoops and try to go fast, because you want to lay a strong foundation. And like me, don't skip math. Math and English are a must, no matter what. Any estimations that I now do, anything, it still involves math. And math and English go hand in hand, regardless of the field that you pick. I don't think it's an option here, but back home where I studied, We had the option to skip math if needed after 10th grade, which is what I did. I would say, hold on to math, it'll really pay off. And soft skills are equally important. Yes, gradually build over it. If there are opportunities in your school to lead a little project or a club, you can use all those soft skills that I talked about and make sure that you truly implement it, because you learn so much. Rather than reading a book about communication, if you can actually pick a topic and go in front of 10 people and talk about it, you definitely learn a lot more. So make sure that you implement some of these things early on, because it just doesn't happen overnight. It has to be built over time. And do project-oriented courses if possible, as much as, I did a lot of theoretical courses, but I didn't understand anything about a DNA until I actually did something in the lab. So those lab experiences and some kind of co-ops or internships or whatever that you can get where you can shadow somebody, those will really edge in your memory in terms of your learning of that particular concept than many books. Even though books are extremely helpful, see how you can implement that in a project, which can actually really teach you what it's about. And it's not possible to take every little thing that you want to learn through a course, but certifications do help. I didn't take a course in biospecimen. I didn't take a course in so many different, even in management. But there are many certifications, and it is so easily available these days with Coursera and so many other ways, where if you're interested in something and you're not sure if you can commit to an entire semester or a course, then take a certification, do like a little summer boot camp, do like a little course to see if that's something you're interested in, because you would at least know if that is your thing or not. So, certifications do help, and learning should never, ever stop. It's not like you learn everything in high school and college and then you just get to implement it. No, that's not how it works in real world. Learning never stops in any career at any stage. I've known of people, even in their 60s, they're still learning something. They're still enrolling in courses because things change, technology change, new things come. And again, not everything can be learned just through textbooks and courses. So read, kids, because truly readers become leaders. A lot of what I did were things that I picked up by reading, either books or somebody's blog or a publication. There's only so much you can do through course that gives you the foundation. But if you really expand your knowledge, pick at something that you like and really find who are the leaders in that area, and see if there is like a blog you can read or interact with them, or some article that they have written. Those really gives you a lot of insights. So inculcate that habit right from the beginning. And of course, most of you would have known about this ikigai concept of, identify what you're good at, what you love to do, and what the world needs. That really gives you a lot of fulfillment, and definitely what you can get paid for. So if you can, put yourself and, identify, these things, and then you can easily figure out a field that you can be passionate about. It may not always be the case, but trust me, in my experience, at least success comes with skill, with some luck, as well as persistence. So if I have the skill and I'm lucky enough to get a project, I still have to persist to do well. And if I'm lucky enough to, get something and I stay, I can build a skill. So they all go hand in hand. It's really important to have the patience. I know some people get a little frustrated when things go south a little bit in their project and they think that, okay, this is not for me. But if they had persisted a little longer, they might actually reap the benefits of all the hard work that they did. So persistence really matters. Really be resilient and stay, and any adversity that you can come across in your project or what you do, take it as an opportunity, because it's not a perfect world. And from the beginning, stick to your core values. Please make sure that you work hard, you're truthful, you have that integrity, include your team, even if it is like a little group of kids working on a project. Really give everyone a voice, make sure that everyone is heard, because you never know, you will learn so much just by listening sometimes. And it's a very important skill down the road when you get into a career, to make sure that you listen and you learn from that, and never ever be afraid to try, because you always learn so much more from trying and failing rather than assuming this is not for me. Because you never know. It'll be very, very revealing when you try it out and see it for yourself. So definitely keep those in mind. And I'm happy to take questions.
All right, thank you so much. That wraps up the presentation. Now I'll be getting into some prepared questions. Most of them were actually answered, so I only have one question that I'm going to ask. After this question, the live listeners are open to ask anything they wish. But yeah, first I'll ask one question over here. So what do you think were some of your biggest obstacles that you encountered and overcame throughout high school, college, job, or any startup experiences, if you've ever been in any?
Sure. I mean, some obstacles, or I wouldn't say obstacles, but I would say those were some missteps. My strong advice for you all to do your education, as in, don't be in a rush to get into the job market. Really make sure that as much as possible, when you have the support of parents, when you don't have to earn a living or put food on the table or take your kid to the school, when you don't have those responsibilities, as much as possible, really push your education up, get as many degrees as possible. Because as you all know, I was so interested in research, but I immediately after my master's took a job. I did start a PhD, but with a full-time job, and like, it was so difficult. It's not impossible, but it really slows your momentum down. And I wish I had like really pushed and finished that PhD, or I had pushed and finished few things earlier before I got too busy with few other things. So as much as it can be very, I mean, if you really think that, taking a job will solidify your interest in a career, then that's okay. But even back in our days, just a master's itself was not enough. And I see a lot of kids do get a job right after they are undergrad, but that's not the bottom line. How far can you go with just an undergrad, right? You need education, and education matters. So as much as possible, keep that clarity in yourselves and never let little things be in the way of getting a good education. And I generally am not a person who would say, you absolutely need to go to Harvard or any of the Ivy Leagues to do well. I think you can study anywhere. It doesn't matter. What matters is how well you do. Yes, Harvard will give you a great head start, but if that's not the program you want, just a Harvard on a program that, doesn't really interest you is not going to get you too far, even though it might give you your first break. Be really passionate about the program, regardless of, what the name brand is. Be really driven to do that. So I would say, not completing my PhD, or, not pushing a little far along in my education is something I should have done a little more back then, even though I felt like, okay, genomics is my thing. As soon as I saw it, as soon as I got a job, I kind of moved forward with it. But given a chance, and if I have to go back, I would have fixed some of these things, because it would have put me in a much better place to understand things better now and perform even better.
For sure. I have one last question before we open it up to the listener questions. Do you think that there are plentiful of opportunities for new opportunities and new business and startup ideas in genomics? And how do you get exposure to those different ideas that haven't been published, like new original ideas for new sorts of cancer research?
Absolutely, absolutely. I mean, I would highly recommend anyone to go to NIH website. NIH is really big on internships. It is one of the biggest entities that really supports helping students. And there are multiple opportunities that you can, I know with COVID, things have slowed down, especially with in-person internships, especially in labs. But if any of you would be interested in data science, analyzing data, there are so many avenues to get publicly available data that you can easily use to get your hands in. I can point you to many who would be interested in guiding you guys. Those are all, really easy things to do. And NIST is an example. They are also very big on standards and internships. And genomics, proteomics is still a very niche, narrow field. You can definitely give that a shot if you want to try to understand the sequencing technology or analyze the genomic data. It depends on what you want to do, but there are plenty of opportunities. I can certainly point you guys to it.
Definitely. Now we'll be going to the questions from the live listeners. So there's one question from Shrudri. Is there an intersection between AI, machine learning, and proteogenomics? What do you think about
Yes,
that?
yes, absolutely. There's a lot of work that is happening in AI, machine learning, as well as proteogenomics. We do what is called a pan-can, where we combine all the cancer types and look for commonalities. And we have applied a STEMness research on it using AI and machine learning. And I'm actually happy to send a little presentation, a publicly available presentation, as well as a publication into your Discord server. If is that Shruti? Yeah, Shruti or anyone else is interested in, can certainly take a look. And if you have more questions, I'm happy to answer them.
Definitely. All right. So for the rest of the listener questions, if there are any, there are two main methods that you can use to ask them. You can either message the questions publicly, you can private message them to the guest speaker or to me, or you can just raise your hand in the Zoom and we'll unmute you for you to ask your questions. So we'll give a couple of minutes now for you to brainstorm any questions that you want to ask. And if there are none to be asked, then we'll go straight to the conclusion and wrap up. All right, so it seems I'm not going to wait an extensive amount of time, but if you have any questions, feel free to leave them in the Discord server. You can take your time for that. I don't really want to rush the presentation, but I'll be going on to the wrap up now. So um, if you're looking for more resources for pathways or more ways to get in contact with the guest speakers, there are a variety of ways. The first step I recommend is definitely join the Discord server. In the Discord server, we have links to the Pathways YouTube channel. And on top of that, we have Google Drive folders for guest speakers, we have a Google Drive folder for members. We have all sorts of good information. We have a calendar for when the next guest speakers will be here. If you want to put that in your bookmark that in your calendars. We have extra additional resources, and soon we'll be opening up leadership opportunities for people to be, working on the Discord server, managing that, or be managers in the Zooms. And we also might start partnerships soon. So those news on those new partnerships and opportunities will also be in the Discord server. This whole entire video right here will be recorded. The recording of this presentation will be posted on the Pathways YouTube channel, and then all the resources I just listed will be in the description of that video. So if there are no more questions, we'll be wrapping up now. Thank you so much, Ms. Mathangi, for the presentation. I've definitely learned a lot, and I hope all of the other listeners did too as well.
It was my pleasure to talk to you all. Thank you so much. And I'm more than happy to answer any further questions and point you to links, books, contacts, and resources. It's amazing what you guys are doing. So I'm definitely happy to be a part of it.
All right. Thank you so much, everyone. Have a nice day.